a dry, sun-scorched land where red dust covers everything. Beneath the exposed stratigraphic layers at the edge of East Africa, once an ancient floodplain millions of years ago, a team of archaeologists begins to dig into the hardened earth. Amid the sediments and debris, a curved structure emerges, not rock, but bone, a fragment of a skull, then another. Piece by piece, they form a strange whole, a nearly complete skull, bearing the shape of something half human, half ape. Potassium-argon dating reveals that this specimen has been here for 1.9 million years. This is no ordinary discovery. The skull predates any previously known Homo specimen found in East Africa, a timeline that significantly pushes back the starting point of modern humans in the region. Immediate questions arise. Who is this? A direct ancestor of Homo sapiens? A previously unknown early form of Homo? Or a parallel evolutionary branch that once existed and went extinct without a trace? The scientific community swiftly turns its attention to the discovery site, a geologically ancient depression near Lake Turkana, Kenya, or the Dmanisi region, Georgia, depending on the specific specimen in question. Here, unique geological conditions, with thick volcanic ash and stable sediment layers, have preserved the pristine remains of a being that lived nearly 2 million years ago. The excavation did not begin with a groundbreaking discovery, but with a routine survey. The archaeologists had only intended to sample rock layers containing volcanic ash, but when they uncovered the first skull fragment with a shape that didn't match any known Homo species, they were forced to stop, expand the excavation zone, and request support from international research institutes. Day by day, the soil was carefully peeled away. Finally, a complete skull was lifted from the ground and placed into human hands, as if the earth itself were returning a long-buried memory. The scientific community's initial reaction was astonishment. Some anthropologists claimed it was a previously unseen transitional form between Homo habilis and Homo erectus. Others strongly disagreed, arguing that its anatomical features did not match any known Homo lineage. Journals like Nature and Science quickly published in-depth analyses, placing the specimen at the center of global scientific controversy. What makes this 1.9-million-year-old skull so special is not merely its age, but the strange combination of primitive and modern traits. Its brain size is small, similar to Homo habilis, yet its flat face and receding jaw resemble Homo erectus, a sloped forehead, but eye sockets that are oddly modern. No single description fits completely. No traditional label can fully explain it. The specimen stands at a crossroads of human ancestry, a hybrid of evolutionary eras. For that reason, scientists don't see this as just another fossil. They call it the missing piece in the evolutionary story, a link that might connect the blurred dots between past and present. Most fossils simply provide morphological data, but this one does the opposite. It disrupts current models and forces humanity to rewrite the opening chapter of the Homo story. So what comes next? How will scientists analyze this skull? And what secrets lie hidden in every morphological detail? The answers await in the next chapter. Part 2. Unearthing Ancient Ancestors. The skull structure indicates an adult individual, possibly elderly at the time of death. The brain volume measures only about 600 to 650 centimeters cubed, smaller than Homo erectus, 800 to 1,000 centimeters cubed, and only slightly larger than that of Australopithecus, around 450 centimeters cubed. The cranial vault is low, with no pronounced forehead, forming a sloping dome that extends backward, typical of archaic hominins. The cheekbones are broad and high, creating a triangular facial profile. The eye sockets are large but not deeply set. The lower jaw is strong, but not protruding like that of Australopithecus. Notably, the molars remain quite large, though not excessively so, indicating that tooth size reduction had begun, a trend consistent with the evolution of the genus Homo. The front teeth are worn, and some had fallen out before death. There are no signs of infection or abscess, suggesting that the individual lived for years without the ability to chew on their own. The cranial sutures are fully fused, a sign of old age. The skull shows no deformities or trauma-related injuries, implying a natural death or one caused by health-related issues. Compared to Homo habilis, this skull has a similar brain size, but a flatter face and smaller jaws, suggesting a transitional phase. Compared to Australopithecus, the individual had a larger brain, more refined jaw and orbital structures, aligning with the evolutionary trajectory toward the Homo genus. On the other hand, when placed beside Homo erectus, this skull lacks key features such as a prominent forehead, developed occipital bone, and larger brain volume. This implies that it appeared before, or at best, contemporaneously, but had not yet fully evolved into Erectus. This specimen sits between two worlds. It doesn't fully belong to Homo habilis, nor does it reach the evolutionary threshold of Homo erectus. Its morphological structure points to an intermediate form, a being situated right at the intersection of evolutionary transition. It may be called a missing link, an ancestor never formally named, but present at a pivotal stage of human evolution. The most striking detail lies in the teeth. Closer analysis shows many teeth were lost during life, yet there's no evidence that death followed soon after. This means the individual survived a long time without the ability to chew food. In the wild, toothless animals are usually left behind and starve. But in this case, the individual continued living. Perhaps others chewed food for them, cooked it soft, or supported them in other ways. This suggests a profound implication. Mutual care may have already emerged, even at a rudimentary level. If that assumption is correct, this specimen represents the earliest physical evidence of organized social behavior, a foundational element in the superior evolutionary strength of Homo sapiens. There were no tools, no fire, no accompanying artifacts, but this skull carries a powerful message. Survival was no longer a matter of the lone individual, but a product of the group. It may have been a small community without complex language or social structure, but it was sufficient for an elderly, weakened individual to continue living for years. This marks a subtle yet vital biological to social transition in human history. Anthropologists have not given this specimen an official name. It does not perfectly match any known species. And precisely because of that, its value is immense. It forces evolutionary hypotheses to shift. It creates a zone of overlap where Homo is no longer seen as a singular product, but as the result of multiple, simultaneous, overlapping evolutionary lines. Part 3. Challenging our understanding. For nearly a century, the classical theory of human evolution has been presented as a linear sequence. From Australopithecus, the bipedal ape, to Homo habilis, the first species to use stone tools, then Homo erectus, the fire-wielding migrant out of Africa, and finally, Homo sapiens, us. Each species was seen as a rung on the evolutionary ladder, a clear next step in a continuous journey. But the 1.9-million-year-old skull disrupts that linear chain. It does not fully fit into any of the established links in that straight axis. Its brain size is similar to Homo habilis, but its jaw and cheekbone structures resemble Homo erectus. Its cranial vault is low like Australopithecus, yet its eye socket placement and nasal bones suggest more modern traits. No simple model can fit all of these features into a single known species. The existence of this specimen forces scientists to abandon the notion of a single evolutionary trunk, where older branches die off to make way for newer ones. Instead, a new model emerges, the evolutionary bush, where multiple species coexist, overlap, branch out, merge, and possibly interbreed. This skull is a physical manifestation of that model. It doesn't belong squarely to one species, but sits at the intersection of several ancient genetic lines. This raises a fundamental question in taxonomy. Are Homo habilis and Homo erectus truly separate species, or are they simply two ends of a morphological spectrum, within which intermediate forms like this specimen are not anomalies, but the norm? If the latter is true, then the traditional naming system, based on a few isolated fossil samples, has likely oversimplified the actual biological complexity. We may be looking at different morphs of one broad species, shaped by varying geography, diet, and survival strategies. Some anthropologists have proposed a new concept, the morphospecies spectrum, a range of physical forms rather than rigid species categories. From this perspective, the transition from Homo habilis to Erectus was not a revolution, but a gradual overlap spanning hundreds of thousands of years, filled with hybrid individuals bearing features of both. The 1.9-million-year-old skull is evidence of an ongoing non-linear, symbiotic process of evolution. It proves that becoming human wasn't a story of replacement, but of accumulation, integration, and diversification. The impact of this discovery goes far beyond archaeology. It compels us to reconsider the very concept of ancestor. If evolution is not a clean succession, then our ancestors were not a single species, but many species coexisting, interacting, and shaping each other. The existence of Homo sapiens is the result of biological complexity, not a linear inheritance. This discovery also reshapes how we understand ourselves. If humans are the product of a densely branching system, then difference is not deviation. It is the very nature of evolution. We did not arrive by following a correct path, but through many detours, collisions, and convergences. The 1.9-million-year-old skull, with its blended features and ambiguous place in the evolutionary tree, does not answer every question, but it forces us to reset the entire framework of those questions. And that is the true power of science, not to confirm what we already know, but to reveal what we had never imagined. Part 4. The Science of the Past. Dating the 1.9-million-year-old skull is not based on speculation, but on rigorous scientific methods. The most common is the potassium-argon (Kr) radiometric dating technique. In volcanic environments, potassium-40 decays into argon-40 at a fixed rate. By measuring the ratio of these isotopes in the rock surrounding the skull, geologists can calculate the precise age of the sediment and thereby infer the age of the specimen. Stratigraphic analysis also plays a crucial role. The skull was found in a soil layer sandwiched between two layers of volcanic ash that had been previously dated. Volcanic ash, with its unique chemical composition, acts as a natural timestamp. If the specimen lies between ash layers dated to 1.85 and 1.95 million years ago, the skull's age can be confidently constrained within that range. Combining multiple techniques helps eliminate errors and enhances reliability. This is standard practice in modern archaeology, ensuring that dating claims rest on solid scientific foundations. Once the age is determined, the skull is scanned using 3D imaging technology. With high-powered laser scanners and X-rays, researchers collect millions of data points to produce a digitally precise model down to the millimeter. This allows reconstruction of the skull's original form, even if it's cracked, broken, or missing some fragments. 3D imaging serves not only reconstruction purposes, it also enables detailed comparison with hundreds of other specimens in global databases. Through micromorphological analysis, such as cranial curvature, eye socket depth, and jaw proportions, paleoanthropologists can determine degrees of similarity with other Homo species. All of this is done using geometric measurement algorithms and statistical analysis, eliminating subjective bias. Another line of research, though challenging, is ancient DNA analysis. Under ideal conditions, small fragments of DNA may remain preserved in bone marrow or microcavities inside the skull. However, with an age close to 2 million years, the chances of retrieving intact DNA are extremely low. Still, research teams are testing ultra-sensitive extraction techniques using enzymes and gene amplification machines in hopes of recovering at least a few viable genetic sequences. This research process is not carried out by a single group. It is the result of interdisciplinary collaboration. Archaeologists handle excavation and preservation. Paleontologists analyze anatomical structures and evolutionary position. Geologists date the specimen and assess the ancient environment. Geneticists hunt for traces of DNA. Each plays a vital role, like links in a chain within a global scientific system. They come from many countries: the United States, the UK, Germany, Kenya, Japan, working together on projects funded by institutions like the Smithsonian, the Max Planck Institute for Evolutionary Anthropology, and the Leakey Foundation. A prime example is the collaborative project between the University of the Witwatersrand, South Africa, and the Natural History Museum in London. There, the skull was scanned, reconstructed, and its data shared with independent analysis centers in Europe and Asia. Results were cross-checked and verified by two separate teams before official publication. These 3D datasets don't just serve current research. They are also publicly archived in digital repositories like Morphosource or Nesbos, allowing scientists around the world to continue exploring and analyzing. The 1.9-million-year-old skull is not merely an archaeological specimen. It is the centerpiece of a modern, precise, and globally connected research system, one that brings the distant past into the light of today's most advanced technologies. Part 5. A legacy of discovery. The 1.9-million-year-old skull is not just a fossil specimen. It is a key that unlocks the door to a series of profound, unanswered questions in human history. It challenges the entire classical classification system. It forces scientists to reassess the evolutionary tree and to reconsider the concepts of species and ancestor within a past that is far more complex and diverse than we once imagined. The research is far from over. The skull has been digitized, and specialized teams are now working on reconstructing the face based on anatomical data. Using jawbone thickness, eye socket dimensions, and nasal structure, a 3D facial model is being built to visualize what this individual may have actually looked like. At the same time, other teams are studying the larynx and throat structures, hoping to estimate vocal capabilities, simulate possible speech sounds, and determine whether this individual could produce simple language-like sounds or merely basic grunts. Another critical line of inquiry focuses on neurological development. Based on the skull's shape, scientists can reconstruct the brain's internal structure, analyzing the size of cortical areas related to vision, motor control, coordination, and social behavior. Preliminary results suggest that although the brain was small, some regions show signs of expansion, indicating that visual cognition and social processing may have been developing much earlier than previously thought. Taken together, this skull becomes one of the clearest and earliest pieces of evidence that human evolution was not a solitary path. There was no single route, no pure lineage. Instead, it was a complex web of coexisting species, competing, interbreeding, assisting, and replacing one another. The story of evolution is no longer about the rise of a single superior species. It is the outcome of interactions among various early human groups, sometimes occupying the same region, sometimes living in the same time frame, and possibly sharing biological ties. Following this direction, scientists are now planning to search for contemporaneous fossils in nearby areas, such as the Homo Valley, Ethiopia, Kubifora, Kenya, or sites in Georgia and South Africa. The goal is to determine whether other similar individuals remain undiscovered, or whether this is a solitary outlier in the evolutionary chain. Meanwhile, another team is developing an ancient migration map by combining fossil records, paleogeological modeling, and traces of archaeological DNA. Their aim is to trace the movement paths of early humans, pinpoint genetic convergence zones, and identify blank regions yet to be excavated. The discovery of the 1.9-million-year-old skull is not the end. It is a beginning, a reminder that we still don't fully understand ourselves. Every bone fragment, every shard of skull, every trace left from the past holds the potential to reshape everything we think we know about humanity. And this story, the one that began in the red earth of Africa nearly 2 million years ago, is still unfolding. What has been discovered is only the surface. What lies unexcavated, unanalyzed, and unimagined may be right beneath our feet. The discovery is not over. It's waiting for the next person to continue the search. It might be you.